Cell counting is a critical task in cell culture experiments, essential for ensuring the accuracy and reproducibility of your results. Although manually counting cells with a hemocytometer is labor-intensive and requires precision, it remains the simplest and most widely adopted method in laboratory settings, particularly when working with a wide variety of cell types. Despite the increasing availability of automated cell counters, manual counting continues to be the most practical approach for researchers. In this tutorial, we will focus on the manual counting method, examining the key techniques and steps that ensure accurate results. We'll also cover the vital role of Trypan Blue staining for assessing cell viability and how to calculate cell concentration based on your counts. Understanding these steps will enable you to perform this task with confidence and precision. To begin, it's crucial that the hemocytometer is properly prepared. Ensure that both the cover slip and the chamber are clean and completely dry to avoid sample distortion. Once your sample is loaded, place the hemocytometer under a microscope equipped with a 100x objective lens. This allows you to clearly visualize the grid pattern of the hemocytometer, where the cells will be counted. The grid on the hemocytometer is divided into several large squares, and your task is to count the number of cells in a specified number of these squares. For consistency, focus on the four corner squares labeled 1, 2, 4, and 5. The counting rule is simple but critical. Only count cells that are touching the top and left boundary lines of each square, but exclude cells touching the bottom and right lines. This ensures uniformity across multiple counts and reduces errors. A common challenge during manual counting is cell clumping. If cells are clumped together, treat them as a single unit, as it is difficult to accurately distinguish individual cells in a cluster. However, if clumps are large or dense, gently resuspend the cells or even resample to break up the clusters and ensure a more accurate count. Uniform dispersion is crucial for precise cell counting. To accurately assess cell viability, we use Trypan Blue, a vital stain. Live cells will exclude the dye and remain clear, while dead cells will take up the dye and appear blue. For accurate results, mix an equal volume of Trypan Blue with your cell suspension and incubate for 3 to 5 minutes. This will allow sufficient time for the dye to penetrate non-viable cells. After counting the cells in the selected grid squares, you'll need to calculate the cell concentration in your original sample. The volume of each large square in the hemocytometer is 0.0001 milliliters. The formula for determining the total cell concentration in your sample is as follows. This formula provides the number of cells per milliliter of your original sample. The dilution factor accounts for any dilution made during sample preparation, ensuring that your final result accurately reflects the cell concentration in the undiluted sample. When assessing viability with Trypan Blue, calculate the percentage of viable cells using the formula. Then, to calculate the viable cell concentration per milliliter, use the factor of 2 is due to the equal volume mixing during Trypan Blue staining, effectively diluting the sample by half. If Trypan Blue staining is not performed at a 1-to-1 ratio, you should calculate the dilution factor based on the actual mixing ratio. In conclusion, manual counting with a hemocytometer remains a reliable and essential technique in the laboratory for determining cell concentration and viability. While automation offers convenience, manual counting provides researchers with the control needed for precise results, especially in a wide range of applications. By mastering the counting rules and calculation formulas we've discussed, you'll ensure the accuracy and reproducibility of your experiments.